Dear students, I am Dr. D. Nagaraju, Assistant Professor of Botany from Government City College, Hyderabad. Today, we are going to discuss about the chromosome structure and function. It is a unit of heredity and inheritance, the very useful part in the nucleus. The chromosome is nothing but the genome. Haploid set of the chromosome is called as genome. You know, the genome chromosome is present in the nucleus. The nucleus is present in the cell, which one it having the nucleolus, nuclear membrane, all the specified nuclear structure that we call as eukaryotes. If it having any specific nucleus, nucleolus structure, nuclear membrane, that we call as prokaryotes. Based upon the nucleus, we divide it into prokaryotes and eukaryotes. In the eukaryotes, specific nucleus is present. These are eye organisms. Prokaryotes are microorganisms, some of the fungi, some algae and bacteria and viruses. Okay. In the eukaryotes, chromosome is present in the nucleus. The chromosome we also call it as nucleoprotein. That means chemically it is a nucleoprotein. Physically it is a bead chain like structure. Pusala danda, bead chain like structure. Physically it is a bead chain like structure. It having the chain like structure, because of the chain like structure only it will be condensed. Otherwise our whole DNA is up to 10,000 kilometers if it is widespread and it is. But it is highly condensed. This highly condensed form of the chromosome is called as solenoid model. In the solenoid model, nucleosomes are present. The nucleosomes, it is a single strand chromatid fiber. It is made up with histone proteins and DNA. Chemically, the chromosome is having the DNA, RNA and proteins. The protein is 40 to 50% and DNA is 50 to 65%, RNA only up to 10%. The basic hereditary material is DNA. DNA is the hereditary material that is having in the chromosome. You see, chromosome structure is varies from one stage to another stage. In all the stages, in all the cell, in all the time, it is not in the same structure. Whenever it is going to interface, that means going to take the further divisions, that time it will be as like a small chain thread like structure. And whenever it is completed the S phase, G and S phase, then whenever it is going to metaphase, that means division phase, it is forming as like a, you can see here the thick chromatid fibers. With the thick two chromatid fibers, each one is called as chromatid fibers. Two chromatid fibers, it has condensed and it has jointed with the specific structure that we call as centromere. Center, it is at the center, it combines the two chromatid fibers, that is why it is called as centromere. You know, there is a very specific changes are there in each and every organism. You can see how the chromosome, where the chromosome, you see here in the cell especially nucleus, in the nucleus chromosomes are present. You see in the condensed type of the chromosomes, dioidic type of the chromosomes, you can see clearly how the chromosomes are in the nucleus. Here, you know, the chromosomes are first discovered by Hofmeister. Hofmeister first discovered the chromosome in the pollen mother cell, that means in the anther, pollens, and Strasburger first observed chromosome in the cell division. Just we have seen in the cell division how it is, in a which stage, how it is, it look like, whether it is thread like or bond like or diode, or it is dividing, where it dividing, what the function it is, this all observed by Strasburger. And Waldeyer given the name it as chromosome. Waldeyer given the name. And after studying of the so many studies of the cell studies, cytogenetical studies, Sutton and Boveri proposed the theory of inheritance. That means chromosomes are participating in the inheritance. That means carrying up the genetic material from parents to upstream. Inheritance. You know, in the genetics means study about the heredity and variation. Heredity and variation. In the heredity, chromosomes are basic units. In the chromosome, whatever the gene, DNA, the DNA is the basic hereditary material that is binded with the proteins that nothing but we called as chromonema. That chromonemata is nothing but the chromosome. And you know the Fleming identified the chromatin fiber. Whatever the chromosome which has taken the stain, that we call it as chromatid. You know, chromo means color, zo means body, color body. The color body, color thread like body is called as chromosome. Okay. This chromosome theory, after the chromosome theory, lot of works have done on the chromosome, on the DNA, and you know the theory, whatever the term, the chromosome, how it is binded like a bead chain. That bead chain like structure we call as nucleosome. That nucleosome term given by Korenberg. Okay. And then next you can see, already told you the chroma means color, soma means body. The color body, it is a rod shape, it take the dark stain in the body. You know the chromosome we stain with the fuelgen, oreacin or estocarmine. Whenever we use these stains, automatically it absorb, DNA will absorb this stain. This will be colorful, we can easily identify this color body in the nucleus. Okay. You know in the chromosome, protein, DNA, RNA, these three are there. These three commonly called it as we call as nucleoproteins, nucleus part and protein part, nucleoprotein. Whereas in the virus, the basic prokaryote also nucleoprotein, we call it as nucleoprotein, DNA and protein. Like that here also nucleoproteins are there. They are having the self replication and it play a vital role in the heredity, mutation, variation and evolution of the species. You know I already told you the 30 to 40% of the DNA and 50 to 65% of the DNA, 30 to 40% of the protein and 10% of the RNA is present. Usually in the G1 phase, the chromosomes are single chromosomes are present, and a stage the chromosomes are two sister chromatids are jointed with the centromere, so that we can easily observe in the interphase. The G1S, G2, 3 phases we call it as interphase. In the interphase only chromosomes will be multiplied. Chromosome multiplication will take place in the interphase. And then in the metaphase, that means division phase, there it chromosome divided into 2 at the centromere part. So that the 2 divided part will become the 2 haploid chromatids. 2 haploid chromatids that goes to the whether the male, female, in the male it is in the sperm and female it is egg cell. So that these 2 divided, these structure, because of this division only, in all the living beings chromosomes are in equal number. Otherwise our chromosome will multiply from one generation to other generation. If it multiply, it cause for the cancer, it cause for the so many abnormal cell growths. Because of this interface, this maintaining the equilibrium of the number, chromosome number. That is why in all the living organism always chromosome number is in same, no change. Okay. And whatever the chromatid, DNA, RNA and histone proteins commonly called as chromatin. Not only histone proteins, it also having the non-histone proteins. DNA is in acidic condition, so that the whatever the basic amino acids arginine that will be joined to this DNA, so that it will be tightly binded. For that binding to form as a bead chain, the histone proteins are very useful as like a binding proteins. These binding proteins, it binds the DNA with the chain like proteins. Okay, that highly condensing packaged DNA will be formed because of the histone proteins. You know the histone proteins are 5 types, that are H1, H2A, H2B, H3 and H4. These are the 5 type of the histone proteins that are made up with arginine and lysine, and also 50% of the non-histone chromatin proteins also present, that are especially actin protein and alpha beta tubulin and myosin proteins. These two are useful to bind tightly the chromosome. You see, the smallest structural component of the chromatid, it tightly coiled with the DNA, histone proteins and DNA, that we calling it as nucleosome. We already said the Korenberg identified the nucleosomes. That nucleosome structure, you see how we have seen the chromosome just in the video. This chromosome is not a single thread, it having the multitask, multi-number, it is highly folded. You see the DNA is the genetic material, the DNA genetic material, it is combined with the histone protein, combined it forming the bead like structure surrounding with the histone proteins. You know, H2A, H2B, H3, H4, these 4 are combined with the DNA, and whereas the H1 protein, it is a linkage protein, it link between the 2 beads, between the 2 rendu పూసల మధ్య, between the 2 beads it interconnect, that is why it is a link protein, so that it form the chain like structure, it form the chain like structure, the chain which are having the beads. Whenever the beads chain like structure, it is condensed, suppose 10 folds of the nucleosomes which form the bead strings, that we nothing but we call it as nucleosome. Here you can see as like a bead chain, పూసల దండలాగా, at the middle here and there as like a beads, it is covering tightly coiling with the DNA, that we look like a bead chain, and when these bead chains will be jointed and folded with the histone proteins, binding proteins. You see here the 4 blue color histone proteins are H2A, H2B, H3, H4. These are binded, and H1 histone protein green color, that is interconnecting in between the 2 beads. That all the part structure with the chromosome packing, whatever the packing chromosome which are there, that again highly condensed, spirally coiled, and you see the 30 nanometer diameter of the chromatid, that means nucleosome, is called as solenoid fiber, solenoid model, solenoid, tightly coiled chromatid, tightly coiled nucleosome is called as solenoid. Because of these tightly coiled solenoids, it will become the very condensed, it can merge within the short span of the areas. You can see the loops like structures also you can see, and you know what we have said, haploid set of the chromosome is called as genome, not the diploid, haploid, that means one set of the chromosome, one set of the chromosome is called as genome, our whole genome. That you know the genomes based upon haploid, haploids are present in the gamete, diploids are present in the sporophyte. That means all the somatic body, in all our somatic body we are having the diploid cells, whereas in the gamete cells, that means in the egg cell or sperm, we are having the haploid, that means single set of the chromosome. That single set of the chromosome is called as genome. You know, where the genes are present in the chromosome, that place we call it as locus, that place we call it as locus, and all the amount of the DNA applied set of the genome of the species is called as C value, that is the total value of the DNA, that is the total value. You know, the chromosome number varies from species to species, but the same species in each whatever the species, if you take human being, always in all the generation it will be the same, but it differ from one species to other species. Okay. Based upon that we can know the cytologically and taxonomically we can differentiate. You know, highest number of the chromosomes are there in Alacantha, that is protozoa, it having the 1600 chromosomes, 1600 chromosome, highest number of the chromosome. And you know the lowest number of the chromosome which having, the lowest number of the chromosomes which are present in Ascaris, Ascaris megacephalaris. You know that it is platyhelminthes. This helminth, it causes the ascariasis disease. That helminth, it having the lowest, that means only 2 chromosomes. 2n is equal to 2, that means haploid chromosome is only 1. And whereas in the plant, you know, the Haplopappus gracious, that is graminae, grasses, in the grasses plant, it having the only 4, that means 2 sets of the chromosomes. That is the lowest number of the chromosome which present in the plants. And in the plants, highest number of the chromosomes where it is present? It is in Haplopappus gracious, Haplopappus reticulatum. The Haplopappus reticulatum, it having the 1216 number of the chromosome, that means 13 number of the chromosomes are present. This is the highest number of the chromosome present in the plant and the lowest number of the chromosome present in the plant. These two are very, very important in the competitive exam purpose. In all the exam, they are asking the questions. Which plant having the lowest number of the chromosome? What it is the genome? Genome means it is a haploid set of the chromosome. Okay. You know, in the plant, chromosome is larger than the animal chromosome. Plant chromosome are larger in size than the animal chromosome, and less number of the chromosomes are present in the animal than the plants. And you know, the monocotyledons having the highest number of the chromosome than the dicotyledons. These the specification are there, and the plant having the largest size of the chromosome, and in the plant monocotyledons having the highest number of the and the large sized chromosomes. You know, the grasses, Haplopappus gracious, that is having the highest number of the chromosome. You know, we can study easy study method is the squash method. That squash preparation method is best method to study, to observe the chromosomes under the microscope. Just in the first slide, I have shown you under the microscope how the chromosome, and the chromosome how the chromatid, in the chromatid how again the nuclear fibers, nucleosomes and solenoids and DNA, how it is look like. We can see under the electron microscope only, we can clearly visible. For the naked eye, for the normal microscope, we can see the allium cepa, that means onion chromosomes are larger chromosome, so that we can see under the normal chromosome, normal microscope. So even in the animals also, salamander and grasshopper having the larger size of the chromosome. So that in the animal, salamander and grasshopper, in the plant, Vicia paba and beans, also in allium cepa. In these two, we can observe the chromosomes under the microscope. This will be look like, we can see visible under the microscope how the chromosomes are. You know, the chromosomes are nothing but, I already told you, it is a chromo means color bonds. It having the color, it take the color with the fuelgen or histocarmine or crocin. If whenever we stain, some part of the chromosome it take I stain, some part of the chromosome is take the very less stain. Where the highest stain is taking, that having the lowest, that means number of the DNA, that having the very less number of the DNA, very less number of the genes. Very inactive part of the chromosome will take the highest stain. Whereas the less stain is taken, that is euchromatin, that having the very active chromosome, active DNA, active parts, active DNA is present. Whereas in the heterochromatin, where the heterochromatin, these are the types are there, based upon that perinuclear or incentral, that again there is a concentral and perinucleus are there. You know, if whenever we are studying the chromosome, the whole set of the chromosome, whole set of the chromosome genes we called as C value. That all the set of the chromosome we can see whatever the whole number of the chromosomes either in the plant or animal, that set of the chromosome is called as karyotype. Karyotype is the whole set of the chromosome, and arrangement of the chromosomes in the diagrammatic manner representation, that means in the sequence based upon the centromere. You know, the chromosome having the two lobes, that is short lobe and large lobe, P and Q lobes. Based upon this length of the P and Q lobes, chromatid length, we arrange geometrically and diagrammatically we arrange. That diagrammatical arrangement from large lengthy chromosome to the short length chromosome, that we calling it as ideogram. That we call it as ideogram, ideometric, ideological arrangement, systematic arrangement as per their chromatin layer, chromatin length, that we call as ideogram. And you know the structure of the chromosome, if you take the structure of the chromosome, it having the so many parts, it has there the whatever the chromatid is a single layer, single thread like structure of the chromosome. That chromatid and chromonematas and chromomeres, centromere and secondary constructions and the telomere and satellites. Along with that, the chromosome, some of the chromosomes which are outer enveloped, chromosomes are outer enveloped with the specific layer, that is follicles, so that it will be preserved, it will be safe. And the matrix also is present at especially at the kinetochore or at the centromere places, so that it having the specific structure to combine the two chromatid fibers. You see, I already told you the chromatid fibers we can see visible in the metaphase. What I shown you in the video, clearly visible the two chromatids with the attachment of at the center attached with the kinetochore or centromere. And the chromonemata means what it is? Chromonemata, at the especially at the propage, chromosomal material become very visible, very thin filaments, very thin, as like very small thread like structure, minute thread like that, we call it as chromonemata. The term coined by Wijduski. Okay. That represents the chromatids in the early stage of condensation. And you know the chromomeres are bead like accumulation of the chromatin, as like a bead chain like accumulation of the chromatin. You know the very fundamental at the starting point, at the middle, that we calling it as centromere. The centromere, it connect the two chromatids. The two chromatids will be interconnected in the metaphase, and in the metaphase anaphase, these two chromatids will be separated with the spindle fibers. The chromatid will be split and two chromatids will be separated with the spindle fibers. And you know the secondary construction, only primary construction is called as centromere. Usually the only primary construction is present, but in some of the plants, some of the animals, secondary construction also present. The secondary construction where it is means where the nucleolus structure, nucleolus formation, there it will be present. It is interconnected, it is jointed with the nucleolus. It having the inactive chromosome part, that is secondary construction, also called as nuclear organizer. This nuclear organizer having the 5.8, 18S and 28S type of the ribosomes and RNA. Very less type of the DNA is present, and only in the chromosome 13, 14 and 21 this nuclear organization is present. Okay. Here you can see the clearly the centromere, it is making division with the spindle fibers in the anaphase. In the sexual organ, to half of the chromosome, to set the half of the chromosome, the chromatid separated at the centromere. You can see here the secondary construction where it is at the nucleolus part, and you can see here the what it is telomere at the ending typical part of the chromosome. Is very useful. If the telomere is absent, the chromosome may be attached, bind to the other chromosome. It causes so many abnormalities, chromosomal aberrations, cancer, mutations. It causes so much of the damage. Cells active very drastically. This causes for so much of the damage. That's why to stop the binding of the one chromosome with the other chromosome at the tip, there is a tip cap like structure is there. That cap like structure is called as telomere. And you know the satellite Satellite means just above the secondary construction. Usually secondary constructions are very less. If the primary construction is common kinetochore centromere, the secondary construction where it is the secondary connection just above the secondary construction, that means nuclear region, above that region we call it as satellite. There is a knob like aperture that we call it as satellite. There it repeats the DNA sequences. That's why we call it as satellite chromosome. Satellite as like a satellite. You know, on the basis of centromere location, chromosomes are divided into 4. If the centromere at the tip only one arm is present, either P or Q arm, only long P arm is present, Q arms are not present. That means centromere at the typical place. That we call as what type it is? Telomere. What type it is? Telomeric. And then if the centromere just below the tip, that means if we write as like an English letter, it will be like a J like structure chromosomes, that we call as acrocentric. And then if the chromosome centromere just above the middle, if you draw the diagram as like if it is look like a L shape structure, L shape structures, that we call it as submetacentric. Usually majority of the chromosome centromere at the middle, if we see the diagram of in the metapage or anapage, it will be look like a V shape structure, that we calling it as metacentric. And also on the basis of number of centromeres, if it having the centromere or not, if the centromere present are monocentric, dicentric, polycentric or holocentric, that means the centromere totally absent, whatever the all surface behave as like a centromere. These are the types of the centromere chromosome based upon the number, based upon the centromere and based upon the number of the chromosomes, and along with that the special type of the chromosomes also present, very big chromosomes also present. Usually the chromosome size is 0.25 millimicron in the fungi, and 30 millimicrons in the plant, and the trillium having the highest size of the chromosome. That means trillium is a very important in the competitive exam. Metapage we can see the largest of the chromosome size, and even in the Drosophila also we can see, and even in our mage 12 microns, and in our human being 10 millimicrons. You can see the based upon the size also, variation also, they divided into 3 special type of the chromosome, that are polytene chromosome, Lambrus chromosome and B chromosome. The polytene chromosome is very gained, large, big size chromosome. It is present in polytene salivary glands, that's why named it as polytene chromosome. These are the special type of the chromosomes, polytene chromosome, which are there in the multi number of the polytenes, multi number of the chromatids, and Lambrus chromosome, which is look like a brush which clean the flask, that's why Lambrus chromosome. And the B chromatin, where the highest number of the heterochromatin, inactive chromosomes, that we call as B chromosomes. Okay. And we also divided the chromosome into autosomes and allosomes. That means chromosomes which are actively participate, which are causing for the makeup of the body, that are autosomes, somatic chromosomes. Whereas the sexual chromosomes which determine the sex, whether male or female. If suppose XX, that is female, XY, that is male. The chromosomes not only always in the single, not only haploid, they are there haploid, diploid, polyploid also present. What we are eating wheat, very high granular wheat, highly delicious wheat, bread wheat, that is polyploid, that means exoploid, 6 sets of the chromosomes are present. And then majority of the functions are there for the chromosome. It is a genetic code storage. It stores the genetic material. All the genetic material is stored in the chromosome, chromatin fibers. That is in especially DNA. and it determines the sex, whether male or female, and it controls the cell division, whether the cell division actually it is happening or not, interphase, metaphase stages are actually happening or not. This all will be decided by the chromosome. If chromosome aberration is there, the cell division multiplies, it causes for the cancer. And formation of the proteins and storage particle proteins, the chromosome DNA is the basic unit. From the DNA to RNA, RNA to protein, that we call as protein synthesis, central dogma of all the cell action, that is main majority of the function of the chromosome. With this we are concluding. In the chromosome we having the very important types of the chromosome. They ask the question, and structure of the chromosome, structure of the chromosome: chromatid, chromomere, chromonemata, satellites, centromere and secondary nucleus, telomere, satellites. These all are the parts are there in the chromosome, the very important structure of the chromosome. And chemical composition of the chromosome also very important, and physical structure of the chromosome also very important. And gained chromosomes are special type of the chromosome, that are three are there: polytene chromosome, Lambert's chromosome and B chromosome. These three are very important examination point of view. And all the nucleosome also very important. What is it, the nucleosome? And in the short answer point of view, genome. What is it, the genome? Where it is? The genome is very important. Where the gene is present, that also locus, where specific point in the chromosome, that are very important in the examination point of view, in understanding point of view, in the cytology point of view, in the genetics point of view, in the physiology point of view, in all the point of view. The chromosome, the gene is a very fundamental structure that we should know. We all know gene means not our wearing genes, we all having genes inside. In our cell we have having the genes. With that genes only we could able to do all the functions. With that gene action only we could able to synthesize the protein, we could able to do all the action. Without the protein we have no action. That all the protein synthesis or all the metabolic, all the sensation, whatever the sensation, that is because of the DNA, that is because of the gene. If the gene changes, gene modification, you know the sexual gene modifications are there, hemophilia, sex linked aberrations, you know, that mean Elizabeth, Rani Elizabeth, there is no males in their whole entire family because of sex linked chromosome. They have, who they have XY, they all will die, only XX will be remain. That is why there is no gent generation, only mother side only females are producing, females are getting survived, males are not surviving. That like so many aberrations, so many changes, so many modifications are there because of the chromosome structure.